I want to move on to talking through phase 1, 2, and into phase 3 clinical trials. And so basically as a therapy process, as a therapy proceeds through phases 1, 2, and into phase 3 clinical trials, I want to talk through the fact that, comparability, the burden increases. And I want to talk about the why. So, John, I'd love to talk through this with you first. What's the why behind that?
Yeah. I mean, early on, there's a lot less at stake, right? When you're entering phase 1, it's effectively a trial designed to assess safety. And so at that point, you've now established at the end of those 10 to 12 patients that, hey, our drug product is safe. We've done some level of dose escalation. And now we want to go and really monitor efficacy. That's notwithstanding the minor bit of efficacy data that most phase 1 trials generate. But at that point in time, if you're ready to make a change in the process or changing a facility, the onus is on the company then to go back and demonstrate that any changes are not going to affect the safety data that were presented or that were developed previously as part of that phase 1 trial. And so it becomes a bit of a headache for those individuals to think about how they're going to go about doing that. The second part is around efficacy, right? So safety and efficacy are going to be recurring themes as we go through this conversation. Efficacy matters less early for the reasons I just described. That is that safety is at the forefront of those early trials. But as you move forward into phase 2 and phase 3, now we begin to demonstrate efficacy data on the clinical side. And now, as you get further and further along, there's a bigger responsibility for those innovator companies to say, okay, now what are we going to do after this change to demonstrate that the drug product is unchanged in such a way that safety is not going to be a concern. And then above and beyond that, the efficacy component is one that becomes very difficult to prove without actually having additional clinical data. And be remiss at this point if I didn't at least describe or discuss potency as it relates to efficacy, because this is we read a lot about potency assays, and there's a real demand to create better potency assays to help predict efficacy. And I think the field is still a bit short on this in terms of what makes the most sense there. But as a general rule, the one thing I would say is that orthogonal assays to demonstrate biology that are associated with the mechanism of action around potency will go a long way with the agency to say that you've done your due diligence around the process change as it relates to downstream biology, and then the implication of efficacy on the back end.
Yeah, good. Okay. And specifically potency assays and efficacy, we can get to that even more down the line in our presentation. So I'm glad you brought that up now too. Michael, I'd like to hear from you. What are your thoughts on, are you in agreement with John? And, just to reiterate, phase 1, phase 2, into phase 3, the clinical trials, the comparability burden increases. What do you think on that?
Yeah, I think John gave a good summary. The way I think about it is, as we move through the clinical experience from phase 1, the requirement, it is expected that the sponsor is gaining knowledge, but also GMP requirements become more stringent as we get closer to a marketing experience, right? So because the population gets larger. For example, stability claims can become more stringent as we initiate multiple clinical sites in the area of clinical manufacturing sites, for example. In the area of analytics, method validation may be required beyond first-in-man studies. So we have to start thinking about that. But if I think about specific points, body of knowledge, right? As we learn more about the product, obviously we're going to learn more about what constitutes comparability. Some tests may be removed, others may be added. Assumptions change with data. Okay. The second one, GMP requirements evolve also, right? It is expected. Early phase, 5 patients, right? Late phase, 500 patients, or Ceramza at Lilly was 3,600 patients. The risk goes higher, right? So more detailed stability claims, method validation may be required. As I said, as experience is gained, you'll almost certainly want to implement process improvements and changes during the life cycle, and every improvement comes with a burden of comparability. There is no process, I don't care what process it is, that you're not going to be optimizing that as you develop it. You may discover new platforms for analytics that because your current platforms can't meet the late stage or commercial requirement, and thus you have to demonstrate analytical comparability of these new platforms. As material supply gets consumed, bridging new lots of reagents or starting materials may come with a comparability burden as well. And then, of course, as I said, analytics change with experience. And an assay that we used initially may be proven to be inadequate, and new methods would need to be added. So I think that if we think about it in terms of the body of knowledge, right? What we assumed yesterday, the sky is blue, right? Today, the sky is blue and there's the moon up there. There's stars that we could see sometimes during the day. Our evaluation changes, right? So, we just have to be on top of that as we move forward with this experience, with the experience of a clinical drug. So.
Yeah. Okay, good. I'd like to publish the poll results quickly for our audience to see just kind of where everybody stands. Okay, we're kind of tied a little bit with considering in-house manufacturing or other or none of the options. So interesting. All right, I want to talk a little bit about, and John, I'm going to ask you about this, just the importance of comparability overall, playing a key role in, unifying that product development. Give us a little bit of color commentary around just sort of the lead up to why it is important, when to get started, sort of that basic information.
Yeah, I mean, at the end of the day, it's essential to demonstrate the safety and efficacy profile of the drug product from today until the entire life cycle of that given drug is the same. And it's, for some of the reasons we've already talked about, and I would imagine for some of the additional reasons that we will continue to describe throughout the remainder of the hour, it's not easy to do that because we don't know what we don't know today. And as Michael's already talked about, there's a lot of reasons from an analytical perspective, from an equipment perspective, process evolution. There are many, many different reasons why we need to ensure that this safety and efficacy profile is unchanged. And so from my perspective, I think the best thing that, if folks in the audience, really what you need to be thinking about is building the most robust data collection platform you possibly can at the earliest stages of your development, of your drug development. Collecting data that you don't think you need today, but you could need in the future when some of those processes change, could be the difference between a 3-month delay and a year-long delay in your drug development platform. So, data becomes king here, and digitizing it is another key component because it makes those retrospective analyses so much easier as